lab today, we will go ahead and pick out dates on when you're going to present. So check those dates, get that done, get that picked out. And I believe there's a quiz. All right. I think I gave you until the end of the day today. That was the acanthocephalins. So yeah, we still have the acanthocephalins set out. Those will be on the lab practical. Next one. We have, what, 5 weeks left? Is that right? 5 weeks? It's crazy. Anyways, Tri-Beta blood drive is coming up. Don't know the date, but it is coming up. So we do have blood drive. We have this parasite symptoms. Those are 2 extra credits. Blood drive, it'll be if you donate, if you find someone to donate for you, if you try to donate and they say no, or you volunteer, that'll be 5 points on exam number 3. And I know if we fill up, sometimes we fill up and there's no open slots. So if you go outside of the hours, you tell them that you're donating for the Tri-Betas at ASU, and you get credit for it. If you sign up through Tri-Betas, I get a list, but you'll also be given like a little sheet that says this is what you do after you donate. You can snap a picture of that sheet, make sure your name's on it, or whoever's name it is that you had donate for you, and then you can email me that, and we'll get added. All right, let's move forward. So we talked about the life cycle. Life cycles, I'm going to say, is fairly simple. There's really only 2 stages in there: trypanosomote stage and an epimastogote. The epimastogote occurs in our insect vector, which is a tsetse fly, and we have different species of tsetse flies depending on species of Trypanosoma that we are looking at. New terms were introduced: metacyclic and procyclic. They apply to our kinetoplast form. So metacyclic is your non-dividing infective form, and then the procyclic is our dividing form. It's entirely extracellular, so kind of make key points there. And then we have this pleomorphy, where we go from a long, slender form of trypanosomote to a short and stumpy form of trypanosomote. And what we see here is that the flagellum length decreases. So long and slender has a fairly long flagella. That's going to decrease in size. It's going to get smaller. And not only that, but our mitochondrion is going to increase in size and complexity as well. And we'll talk about why that is, why that happens. And it's actually, this is the reason why we start switching to this stumpy form. Once our insect gets infected, takes that stumpy form, then it's about 3 to 4 weeks or so before they are now infective, and they will spread it. All right, any questions on that life cycle? All right, so why transition? So the stumpy form only appears at the peak of the parasitemia. So the parasitemia is the phrase that basically says how many, or it's a quantifies how many parasite individuals are in the host. So high parasitemia, you have a very large number of parasite individuals in the host. Low parasitemia is a very low number. So if we were just to go ahead and do a graph of total parasitemia, and we'll just do N as our sample size, we have time, you're looking something like this. And then we'll talk about why it drops off. But this is our total parasitemia. That stumpy form only starts to appear when we're at very high numbers. All right, so there's probably something that triggers it where it can identify high densities in the host. And then that says, okay, we're at a high density. Some of our trypanosomotes are long forms that are replicating. Some of them have to start differentiating to our stumpy form. So What's that trigger? Well, we know it's a density. Is it something with a parasite or is it something with the host? I don't know. I would guess it has something to do with the host. Because once you start getting these high numbers, the host immune system takes off. And as we're going to see, this drop here is due to the immune system. That's a real crappy piece of chalk. And we'll talk about that immune system. So what's going on? Why is it switching to the form? Well, it's tied to the metabolism of these guys. All right, that's what it's tied to. So when we're in the slender form, we are rapidly dividing. We're in the blood, we're in plasma. All right. We have an abundant source of food, glucose. Blood glucose levels course through there. All right, so we can get by, or the parasite can get by with just glycolysis for all of its ATP production. All right, and if it's going to get by with glycolysis, then it doesn't need anything for the oxidative phosphorylation. It doesn't need the electron transport chain. If we don't need the electron transport chain, then we don't need the complexity of the mitochondria where that is going to take place. So if you go back to your cell bio, if you had it, right, the electron transport chain is relying on the membrane of the mitochondria, right, your double membrane. So you have all of these folds, all the cristae inside of them, increasing surface area of those membranes, so you can have a lot of this stuff going on. Well, in our slender trypamastogote, it doesn't need that. So our mitochondria, our single mitochondria, is going to be very simple. It's going to be very simple, meaning it has a few cristae and that's it. But then we have to prepare for life in the insect vector. So we're going from blood with circulating glycolysis into an area where there's much less nutrients. There are fewer food sources for these guys. And if you have fewer food sources, then you're going to have to do something to increase your production. And one way in which you could do that is just increase the efficiency of ATP by utilizing the electron transport chain. All right? So, you ready for this? So once we start transitioning to the slender form, or from the slender form to the stumpy form, where's my cursor? What's going to happen is the mitochondria is going to start to enlarge and increase in complexity. As that's enlarging and increasing in complexity, it's going to take up a bigger portion of that parasite body, of that trypamastogote form. As it's enlarging, it's slowly pushing the kinetosome anteriorly, so pushing it forward, to the point where when we are in the insect host and we are utilizing electron transport chain for our ATP production, it's pushed it so far ahead that now the kinetosome is anterior to the nucleus. We still have our flagella, we still have the undulating membrane, but now our kinetosome is anterior to the nucleus. What's the name of that life cycle stage? Our epimastogote. So our trypamastogote is where our kinetosome is posterior to the nucleus. Our mitochondria has enlarged, become so complex that it now moves the kinetosome anterior to the nucleus, and that is what marks our new epimastogote stage. Thing is that we're in the insect gut, we're in the salivary gland. Limited oxygen, limited nutrients, still some oxygen, but that means we're going to try to be as efficient as we can, and that means utilizing oxidative phosphorylation for our ATP production. That's the reason why we're transitioning. It's this need to increase efficiency of energy production. And it does so by increasing the size and complexity of the mitochondria, which consequently pushes our kinetoblast forward, our kinetosome forward. Our kinetosome. All right? That's why it transitions. Energy efficiencies. And then once we transition back to the trypamastocyte form, it's starting to degenerate. The mitochondrion is becoming much more simple because it doesn't need it once it gets into the host. And you're transitioning to a trypamastocyte form that's non-dividing. So it's not going to use a whole lot of energy. So everything becomes simplified and remains that way as long as we can get by with just glycolysis. All right, ready? All right, pathology of this. Pathology is primarily against our slender trypamastocytes. That's going to be our dominant form. And we're going to see kind of a sequence of events that happen. First one is that you often get a canker or a sore at the site of infection. So the insect, the tsetse fly, bites you, and you get a sore that persists for about 1 to 2 weeks. That's a little different, right? If you get bit by, by a mosquito, yeah, you might have that little round, raised bite, all right? But it normally resolves itself in a couple of days. You get bit by one of these flies, these biting flies, it's going to be a little bit more serious. This sore, it's from the fly, tsetse fly, and it's also from the immune system. So your trypamastocytes are there, they're going to be replicating. You'll get inflammatory response at that point. But then as it's replicating and your body's starting to generate a big immune response, you'll develop something called winter bottom sign. It's the swelling and congestion of the lymph nodes in the neck, leg, and groin region. So again, no big surprise when you're fighting an infection, your lymph glands, lymph nodes start to swell a bit. All right, this is very characteristic. And it was named, Winterbottom sign was named for a naval officer, British naval officer, who recognized that individuals that had these symptoms probably had a disease that wasn't going to allow them to survive. And it's pretty disgusting what the history was. This was a naval officer. These were slave trade, the slave trade that was happening. So they'd see the individuals exhibit this stuff. They said, well, I'm not going to get my money out of these because they might not survive. Or if they do survive, I'm not going to be able to sell them. So they threw them off the ship, save the resources. And that naval officer, Winterbottom. And it's persisted, the sign, Winterbottom sign, the terminology has persisted. I like to include it because we've had a terrible history with this. So you get swelling. It's all from inflammatory response. And then also with this swelling, you'll start to get these intermittent fevers. Now this is a little different than malaria. So we'll talk about the paroxysms of malaria that happen. This is intermittent fever that is associated with generalized pain, headaches, weaknesses, and so forth. And it's intermittent. You'll get it, and then it'll resolve, and then it'll come back, and then it'll resolve, and it'll come back. And it's going to be tied to our immune system. So I already kind of gave you a preview that our parasitemia shoots up. Our immune system is very active. It gets to the peak, and then it takes out our parasite that's in there. All right? So during this stage, as we go down and stay depressed, now our symptoms start relieving themselves, or they start relaxing until our next wave happens. All right, and you do have possible death. Possible death from this. With Rhodesiense, it's going to be much quicker. Oftentimes you get this, you're going to see rapid weight loss because individuals are no longer eating, and then oftentimes you get the heart involved with it. So it could be some sort of myocarditis, inflammation, your heart just is starting to get stressed, and maybe even cardiac shutdown. But that's not all. And if this stuff isn't bad enough, what else? You can develop this African sleeping sickness. And this isn't necessarily for both of our African trypanosomes. This is just for Gambiense. So African sleeping sickness is this fatigue that's caused by invasion of the nervous system. So the parasite now, instead of just being in the blood and plasma, will get into the central nervous system, and then start to cause this fatigue, this excessive fatigue. Often develops 3 to 4 weeks after infection. This is gambiense. Right. And what you would get is after you already have your winter bottom sign and you're starting to get these periodic or intermittent fevers, right? You're going to start getting increasing apathy, disinclination to work, mental dullness, reduced coordination, just kind of blah. I don't feel like doing anything. And this is kind of tied to increased sleepiness. Now, number one, why is this a good thing for the parasite? Why would that be a good thing for the parasite? Anybody? Who wants to take a shot at it? So they're not moving much? And why is that a good thing? Less likely to die? If left untreated, we will die. But what has to bite them? It's our fly. So as we talk about adaptations, pathology could be an adaptation for successful transmission. It institutes it, and you're right. This isn't just like a rapid death. You're going to have this, and it's going to slow you down, really make them lethargic, not moving, makes them much more likely to be getting nailed by a tsetse fly and thus transmit. So this kind of helps prolong the time which our individual is transmitting. After this, then you can start getting more of the classic nervous system type pathology, which is tremors in the tongue, hands, and trunk. So basically, shaking, loss of control of muscles, and that would be followed by paralysis and convulsions. And then coma and death is usually the last phases of this. And this could take 2, 3, 4 months. This is pretty rare in rhodesiense. So Trypanosoma Cruci rhodesiense. It's still a trypanosome. It could still invade the nervous system. However, rhodesiense is much more severe infection, and the host usually dies before we ever develop this African sleeping sickness phase. Once we get to African sleeping sickness, prognosis is pretty poor. All right, so if you can recognize you've got a canker, you're starting to get the swelling, you're having these intermittent fevers, you need to be checked out. You need to get the drugs that start to combat this parasite. Because once you develop into this African sleeping sickness, it's going to be hard to treat. Drugs that are out there, arsenic-based, all right? About 10% of the individuals that take these drugs will die just from the drugs themselves. Which is like, oh my goodness, why do that? Well, it's kind of a last-ditch effort. Parasites are good. They're good at what they do. Ready? All right, immune evasion. This is really the reason for the intermittent fevers. All right, so how does it do this? Well, we have to recognize that these parasites are in the blood and the tissue fluid. They are facing the full assault of the host immune system. They're dealing with complement-driven lysis. They're dealing with antibodies. They're dealing with cellular responses. All right, they're facing the full force of the host immune system. Yet they persist. They're really good at persisting. So how do they do that? Well, as we said, parasitemia peaks, goes up, all right, and then it drops off. As the parasitemia is picking up in number, what we notice is that this peak now represents a single population of trypanosomes. That population exhibits something called the variant antigen type, or a VAT. The variant antigen type is basically what your immune system is going to recognize. The variant antigen type is determined by the variant-specific surface glycoproteins, the VSGs that are there. These variant surface glycoproteins are exactly that. They are glycoproteins, but they could vary. Now, In our population, you're only ever going to exhibit one type of VSG at any one time in our population. That's not 100% true, but we can say 99% of our parasites have this one VSG, because even at 1%, you're looking at a pretty decent number. I mean, that peak, we're not talking about 100, 200 trypanosomes. We're talking potentially billions of trypanosomes circulating in the blood. So almost all of them are expressing this single VSG. All right. This VSG is a quantitative trait. So if you've had genetics, you probably have been introduced into this. Just kind of as a quick overview, when you learn genetics, when you learn like brown hair and blonde hair, or for the pea plants, right, green versus yellow, or smooth and wrinkled, right? Those are discrete traits. You have your classic 3-to-1 ratios, your 9-to-3 to 3-to-1 for your dihybrid crosses and so forth. Those are very discrete. When you have a continuous or quantitative trait, what you have is multiple loci that code for that phenotype. So height for us is a great example of qualitative trait or quantitative trait. You have many different loci that determine how high you'll be. Well, that's the same thing with the VSGs. You've got easily 1,000 genes that go into coding this specific VSG. And they're in the telomere region, which seems strange, right? But if you're in the telomere range, you have a lot of chances of rearrangement that's happening, producing new combinations of those VSGs. So what happens is you have only one VSG expressed at any one time in our population. That picks up. Our host immune system learns that VSG, produces antibodies to it, and then takes it out. But you had a very small number of trypanosomes that had a different VSG expressed. One of those will now become dominant, and they take off. They can take off and survive because our immune system only recognized this first VSG. So now we have the second wave. Our body has to learn that VSG. It learns it, it takes it out, but that allows another VSG type to become dominant in our population, and it proceeds that way. You get each new VSG happening with each wave. All right. This VSG gets expressed starting at the metacyclic trypanosote stage. So you have your epimastogote in the salivary gland of our tsetse fly. All right, and then it's going to transition to the metacyclic form and detach. So now it's free in the lumen of the salivary gland. That is when it starts exhibiting these variant surface glycoproteins. Why not before? Well, they don't need it. They only need it when they get to that vertebrate host. So they're going to start prepping. They're going to make these things. They actually originate in the flagellar pocket. That's where they originate, and then they spread throughout the body. All right, so you have all these different VSG types. We've got quite a few VSG types, and there seems to be some sort of progression. It's probably some sort of non-random progression that's tied to how the rearrangements happen. But we know a lot about these rearrangements and why they happen, why they occur, and this is one of these things where those telomere regions, they don't keep shrinking like you would have learned. So there's been some research to try to figure out why does it stay the same length, or why does it expand? What are those things? So we're not going to talk about it. That's for like a seminar class or something like that. Any questions? They're going to keep doing this until the host eventually dies. All right, so why do they confer protection? Well, we already talked about this one, periodic antigenic variation. That's the reason for the switch from VAT type 1 to type 2 to type 3 to type 4 and so forth. So we're just periodically changing our surface proteins, and that means that as soon as we change it, we escape the memory response. And then the host has to go through that whole process of learning another memory response. All right? But the other component to that is that these variant-specific glycoproteins, actually stick out from the surface of that worm, or that trypanosomic, it's not a worm, of that protozoan. And because they stick out, they shield all of the other conserved epitopes that are located right near the base of that membrane. So you do have conserved epitopes, or you do have conserved proteins on those membranes. It's those proteins that people have targeted to try to create vaccines for them. But what good is a vaccine if our antibodies can never reach them? So the protection is twofold. One, it shields those conserved regions so that the host, only sees those VSGs. And number two, the changing of the VSTs confer this antigenic variation. That's how we get our protection. Ready? Pretty cool way in which they avoid the host immune system. All right. I'll have to get this quiz up and posted, but we'll probably have this later this week. Set the deadline later this week. All right. So that's the African trypanosomes. Let's talk about the American trypanosomes. All right, so we're going to do the American trypanosomes and then talk about leishmania, which is also kinetoplast. What they are. All right, American trypanosomes. There's one species that we're going to talk about. It's Trapanosoma Cruci. It was first described by a Brazilian physician, Carlos Chagas. All right, that's how the disease got its name. It's Chagas disease. We find it in South and Central America. It extends into the Rio Grande Valley region of Texas. We find Trypanosoma cruzi here in San Angelo and other parts of Texas. But incidentally, even though we find it in the rejuvids here, and even in like San Antonio region, you find it, you don't see the human cases. And I think there's been some studies suggesting that, yeah, it's being identified as Trypanosoma cruzi, but maybe it's some sort of different genotype that can't infect humans. All right. So South Central America extends into the Rio Grande Valley here in Texas. This parasite infects 8 to 11 million individuals annually. It infects a lot of different animals too as reservoir hosts, so 100 to 150 species of animals. Fix that. The vector is a rejuvid bug. It's also called a kissing bug. It infects species in the genus Trypanosoma. We only find Trypanosoma cruzi in the gut of this vector, not in the salivary gland. It's only in the gut. It's going to switch to, it's going to become infective while in the hind portion of the gut. So this is stercorarian transmission, and we already looked up what that root of sterco is, right? Excrement. So this is what I would call our bite and crap method. And you can see it. Rejuvid's feeding, its abdomen swelling, it'll swell up and it'll force out any feces that is in the hind gut of that bug, and in the process deposit any Trypanosoma cruzi that's there. Ready? Not yet. We picked up some rejuvids down at Big Oak River Ranch on the Devil's River, and we did gut smears. I think we picked up about 10 of them. Like 3 or 4 of them we did alive, like gut smears. every single one of them had these trypanosomes inside of it. So, pretty cool. Ready? All right. Life cycle. This one's a little bit more complicated. It's a little bit more complicated because Trypanosoma cruci is an intracellular parasite. Use my new chalk. Let's see how this one works. All right, Trypanosoma cruci. It's like a very soft piece of chalk. All right, so I think what we're gonna do is split it up like we did the other time. So inside our insect vector, we have our metacyclic trypamastigote. This is in the lumen of the rectum. This is our rejuvid. Kissing bug. And I'm gonna put mammal. So it goes into humans, causes some pretty significant pathology in humans, but we can find it in rodents, we can find it in raccoons. That was something I was talking to Dr. Upham about. He's interested in like zoonotic diseases, possibility of surveying some of these small mammals around here. But we have our fight and crap mode of transmission, Stercorarian transmission. That'll happen, or you could actually accidentally consume the bug. So we have our trypamastigote. That's going to be in our blood form. All right, blood and tissue fluid. I guess I should say blood and tissue fluid. All right, and the first thing that's going to happen when we get in there is that this trypamastigote is going to be phagocytized. And I'm going to put phagocytized in quotation marks. All right, it's going to be phagocytized by macrophages that are coming in to respond to our infection. Now I put phagocytized in parentheses because there's actual multiple processes that will bind and then internalize the parasite. All right, it's not just a single pathway. This is what happens. There's multiple pathways that ultimately end up with our trypamastigote. In a macrophage in something called a parasitophorous vacuole. In a parasitophorous vacuole. What this vacuole is just, I mean, that's all it is. You've been internalized, you've been phagocytized. You would call it, what would you call it? An endosome, all right? But this endosome has the parasite, so we call it a parasitophorous vacuole. We're in that vacuole, and then what's going to happen is that we will transform into an amastigote. All right, so an amastigote. No flagella. We have our kinetosome anterior to our nucleus. All right. So I left off epimastocyte, promastocyte phases because we just proceed directly towards that transformation to get to the amastocyte stage. All right. And then what we will have is binary fission and cell lysis. So binary fission and cellular lysis. Binary fission and cellular lysis that causes the macrophage to rupture that releases the amastocyte back into the blood and tissue fluid. All right? Now, with this parasite, there's two things that could happen, and both of them are probably likely. One, these amastocytes now get phagocytized back by our macrophages and other cells of the reticuloendothelial system. And we'll call that our reticulotropic strain. So other macrophages come around, they pick it up, they internalize it. We're already the amastocyte stage, so we don't have to transform, and it continues. Binary fission, cellular lysis, replicates, and so forth. All right, but the other one is that sometimes these amastocytes will invade muscle cells. It's not just striated muscles, it's muscle cells. So it'll hit striated, smooth, and cardiac muscles. You will have your replication and lysis, replication and lysis, releasing the amastocytes back into the blood and tissue fluid, and then these will end up continue replicating in our muscle cells. This is the myotropic strain. So you'll have both. You'll have both of them in the life cycle. Some of these amastocytes, when they get released in the blood and tissue fluid, they will transform back to a trypanosomastocyte. Because that trypanosomastocyte is what gets picked up when a rejuvenate comes and feeds on you. All right, so any questions in our mammal? Hopefully you're starting to see. Once we go into the myotropic strain, there could be, and notice we have cell lysis in both spots. We've got some places for pretty significant pathology. All right, so we have our trypanosomastocyte. This is in our gut. Mid-gut here. All right, we get to our trypanosomastocyte. We then transform to the epimastocyte stage. Mid-gut. And here, our epimastocyte will be replicated by binary fission. Some of these epimastocytes then will migrate and attach to the epithelial cells of the rectal gland. All right, this is our hind gut. Now, with Trypanosoma Cruci, we migrated up to salivary gland. We attached, transformed to epimastocyte, and then you had a couple rounds of division, and then that's it. Then you had the transfer. This does not happen. We get to the rectal gland, we attach, we hang out there, we do some developing, and then part of that developing is transition to a metacyclic trypanosome. And this is attached. And then once we complete transformation to that metacyclic trypanosote stage, then it will detach to where our parasite is now free in the lumen of the rectum. Just to kind of, again, clarify, our entry occurs into the wound. We're not injected. Feces gets deposited. We have our metacyclic trypanosomes in that stage. We have to get to that open wound. Once we get into the open wound, then you're going to have the cellular response coming right to that site. So the first responders, macrophages. Who does the parasite want? Macrophages. The parasite capitalizes on our own bodily process to get the cell that it needs, to phagocytize it, and then it transforms into an amastocyte where it's now going to be replicating. Questions? Ooh, I did not enlarge it. There it is. Enlarge it so you can see it. All right. So, got lifecycle, parasitophorus vacuole, endocytic vacuole that contains a parasite. It fuses with the lysosomes and other endosomes. So normally when you have phagocytosis happening, you get that endosome forming, and then you have the endosome lysosome fusing, all right, where you're dumping enzymes inside of it. You're starting to change the pH of that endosome so that enzymes can break it down, and so forth. And that's really what's kind of happening. All right, so here we've got just a general diagram to show some of the different ways that you can get to this. So you can have a lysosome initially take it in, or you can actually have an endosome and then fuse it with lysosomes to produce this. And what you get is transformation to the amastocyte stage. Amastocyte's going to replicate inside the cell itself. It's going to replicate and then cause that cell, that macrophage, to burst, or lymphoid cells, possibly. As we said, the reticulotropic strain, that's just going to basically stay right with our macrophages. The myotropic strain will infect our muscle cells. So there's a lot of information on how the entry is happening. So if you're interested, go take a look at it. A lot of different signaling pathways. Note, replication occurs in the cytoplasm. Ready? Talk about some of the pathology. All right, so with Trypanosoma Cruci, you get that canker that develops right at the site of infection. Same thing happens with Trypanosoma Cruci. What you get with this is, instead of a canker, you get what's called a shargoma. named, again, after like Chagas disease, right? So it's localized redness and swelling at the site of infection. Also progresses to the regional lymph nodes, so you have inflammation that's happening. You've got the macrophages responding. You've got the parasite that transforms and replicates and kills the macrophages, which calls in even more, so you get this localized swelling, redness and swelling right at the site. If it happens on the face, where it oftentimes does happen at the face, you'll get that same swelling, but now you're getting swelling normally around the eyes, because rejuvids are drawn to some of the CO2. So when you're sleeping, they can nail you in the face, and then you get something called Romagna sign, which is a accumulation of fluid, so edema, in the eyelid and conjunctiva, swelling of the preauricular lymph nodes. You get this, gonna look like you went, 30 seconds with Rocky Balboa, maybe? Say 30 seconds, but you start getting the swelling, and it's in the face. This is kind of a severe case, where it's really swollen. Other images are gonna be less obvious. So, got a lot of different shibgomas images. This one's a bit less. Still, you get swollen eye. That's a bad one. It's again just kind of a swelling that develops, and normally that's where they get hit, where the rejuvid bites you. You get that localized swelling. All right. So shigoma, Romagna sign. That's all Trypanosoma Cruci. We also have this is like the first time we've encountered something where you have two different pathways inside of our host. And those pathways were the ones that stayed with the macrophages, and then you have the other ones that go into the muscles. When you have the muscle cells, when they get infected, that amastogote's still going to replicate and still going to cause the individual cell to rupture. All right, and that's going to produce quite a bit of pathology. So what we see in this is that you tend to get more parasites, more amastogotes in the cell before it ruptures than if it's in a macrophage. That congregation of parasites is called a pseudocyst. All right? So you could say it's a cyst, but cysts imply that it has some sort of cell wall or some sort of thick outer wall covering. That's where the pseudocyst name comes into usage, because it doesn't really have that big thick wall. It's just it's a pseudocyst. It's a cyst. It's a large congregation without that very thick outer wall. When you have this is a great pseudocyst. All of these are amastogotes inside of them that have replicated. When this thing eventually bursts, which it will, it's going to cause a local inflammatory response that's going to cause degeneration and necrosis of surrounding cells, because that's kind of how our body responds. All right, when you have that, what you're doing is basically potentially, or you have the potential to kill off nerve cells that were innervating those muscles. And if you kill off those nerve cells, then the muscle's no longer going to be able to contract. If the muscle no longer contracts, then it's going to start to lose its tone. So you lose muscle function and muscle tone. The end result just kind of depends on the site of infection. So if you're in the heart, and this is a common place for Chagas disease, when it goes to the heart, it can lead to heart failure. And actually, this is typically what you would find when you have individuals, 20s and 30s, that are dying of cardiac arrest, heart failure. First thing people look at is, okay, is there some pollution, which I don't know why you look at that. But then the next thing is, okay, is this a parasite? Is this Chagas disease? And that's kind of how we've identified that there's a hot spot in El Paso, where you just had an abnormally large number of individuals dying of heart failure. All right? If it's in the GI tract, you have the same thing. Again, smooth muscles in the gut. Smooth muscles are moving the food down there. Well, if you stop stop its function. So that's what a normal small intestine would look like, or large intestine, colon. That's what a normal one would look like. When you stop contracting, you start to lose its muscle tone. And if you stop contracting, now you're relying on other things, other parts to push the food through, and you start having expansion, gradual enlargement. So you can get to something called megaesophagus if it's in the esophagus, or megacolon. Or you get really large inflated colon, just drastic enlargement. All right, and this is one. This was at megacolon. Was that autopsy of an individual? That's just massive. So we'll pick this up. We have to talk about host evasion. We'll review the pathology again, but we'll do Chagas disease and then finish up Leishmania on Wednesday. I don't know if we'll finish with Leishmania or not, but we'll at least start it. All right, have a good one. How's it going?
Can I go ahead and get first dibs for my species presentation on the 18th? Just because I do not want to go next week because I have a
Do you have a pen?
test. I do.
We already have someone signed up for the 18th. That'll work.
Perfect. I just have cell bio test next week, and I was like, I do not want to do it on the same week. Thank you so much.
Is it on Friday?
Yes, next Friday.
It's Good Friday.
Yep, I know. I saw that and I was like,